Alright, this is the part 2 video for the Recombinant DNA Technology lecture. This will be brief, there are only 3 slides left. So the first thing I want to talk about is some of the limits that we have using this technology. There are some that are technical, there are some things that we just know are possible somehow, but we haven't been able to do them yet. And then there are some things that are uh, ethically limiting what we do actually use these technologies for. We could do some things, but people have generally agreed, the international community has generally agreed that it's just not a good idea. It's just not okay to do certain things because the technology is so powerful. One of the things that we have made with recombinant DNA technology are super crops. We could make plants that are just super resistant to diseases, but that would potentially give them the ability to overgrow native species. Uh, these super crops could potentially sort of get out of our control and take over whole ecosystems, and that would be very, very damaging to biodiversity. Another thing that we could do using the same basic technology is take genes that give bacteria some antibiotic resistance or some increased virulence, meaning we could change them so that they are more likely to spread from one person to another, or just more deadly, or all of the above. We could make diseases that are very, very difficult to treat, very, very, very deadly, and very easy to spread from one person to another. We would call those super germs, and we have the ability to make that right now, but we, for the most part at least publicly, have decided not to. Another thing that we have decided not to change using recombinant DNA is human embryos. We're okay with moving genes from humans into something like E. coli, but again, we have generally agreed as a global society that we are not going to be moving genes from other organisms into human embryos and then growing those humans up with the other organisms' DNA in them. So we really need to decide, how much are we going to change nature? How much are we going to potentially change ourselves? And we have to understand that this technology does hold a huge, huge potential to help us, but there is some real danger to using it as well. As far as the technical limitations go, for right now, we really are just moving a single gene, a single protein, and that's pretty easy to do. But more complicated things involving several genes, we just haven't really figured out how to do yet because we don't have a great understanding of how genes get regulated. I mentioned all that used to be called junk DNA before, and we know that the non-coding DNA plays a role, but we don't quite understand exactly how it works, so we can't really control it very well, and we're still a long way away from being able to do something more complicated than just moving a single gene from one organism to another. So the idea that we're going to suddenly be able to grow like whole body parts or even just tissue and organs using this technology, we're not there yet, and we probably won't be there all that soon. We are starting to overcome some of these technical limitations. One of the relatively new discoveries we've made are the existence of genes that seem to regulate a whole bunch of other genes. That some of gene regulation does actually occur, not in the non-coding DNA, but in the coding DNA. One of the first discoveries was this group of genes. They're called Hox genes in fruit flies, and we figured out that this one gene controls a, a whole bunch of other genes, and if the wrong Hox gene gets turned on in the wrong part of her fruit fly's body, it can do something like grow a leg where it's supposed to grow an antenna. There's one Hox gene that turns on all of the proteins that make an antenna. There's another Hox gene that turns on all of the proteins that make a leg. And if you turn on the wrong one, then you end up with a fruit fly with two legs growing out of its face. So we are beginning to understand how we could do a better job of regulating not just a single gene, but whole groups of genes. And something we do kind of need to ask ourselves is should we keep going with this? Should we get to the point where we could like grow eagle wings on a horse or a person? That's obviously very far in the future, uh, but it's something that we should start thinking about now. So over the next week or so, we'll keep talking about different ways that we could potentially use biotechnology in the future. And I'll try and give you some framework of sort of how we decide, okay, just because we could, does that really mean that we should? Uh, so to complete this assignment, I just need you to submit your notes to me. You can do that. You can attach them to an email, put them in a body of an email, or just share a Google Doc. I just need to see that you watched both parts of this lecture. Enjoy the rest of your day.